تمام، هيك مبين عندكم واضح؟
دكتورة.
تمام. اوكي. طيب, so today we'll start with the topic of carbohydrate metabolism. It's actually going to be just an introduction and some review of carbohydrate as a topic. We're going to talk about the digestion and absorption of carbohydrates, and your reference, definitely the main one, is Lippincott Illustrated Reviews for Biochemistry. Okay. The topics in carbohydrate metabolism generally that we're going to cover مش today, يعني في كل البلوك راح يكون utilization of glucose, يعني عملية glycolysis, in which glucose is broken down into smaller molecules, so the energy inside it can be extracted and used to perform the different physiological and biological functions. راح نحكي عن synthesis of glucose from other sources أو non-carbohydrate sources اللي هي عملية gluconeogenesis. We're going to talk about the storage of glucose once we have excess of it, اللي هي synthesis of glycogen, and we're going to discuss the breakdown of glycogen into glucose to release it and use it as a source of energy under certain conditions. We're going to discuss also the pentose phosphate pathway that is used to produce pentoses as sugars, as well as NADPH, the reducing power. And we're going to talk about the metabolism of other monosaccharides and the interconversion between different sugars. So let's start first with defining sugars. If you remember, in the last semester, we discussed the carbohydrates, and we said that they actually belong to the aldehyde or ketone family, and in addition to the carbonyl carbon they also have hydroxyl groups, so they're polyhydroxyaldehydes or ketones, and so they're so-called aldoses or ketoses. Okay, so the aldehyde group, as you can see in here, is the terminal one, is the first one, so it has to have number 1, and the simplest aldehyde sugar or aldose is glyceraldehyde, the one you see on the left of the screen. This one on the right, dihydroxyacetone, is the simplest ketose, made again of 3 carbons with the carbonyl carbon in the middle, or at the second carbon, number 2. Okay. The same thing applies if we add more carbons, not just 3 ones. If we have 5 carbons and we're talking about aldoses, it becomes ribose. If it is the ketose of ribose, or the ketose that is similar to ribose, is ribulose. 6 carbons or hexoses, again aldehyde is called glucose, and the same one with the same formula but different distribution with the carbonyl carbon on position number 2, this is called fructose. Here is a group or examples of different types of monosaccharides that we can find in our cells, but the main ones are actually pentoses and hexoses, made of 5 carbons and 6 carbons respectively. So glyceraldehyde is an example as a triose, erythrose is a tetrose, ribose, as I said, is a pentose. There are other types as well. There's the glucose as a hexose, galactose as well, but glucose is much more common. The heptoses, an example on them is the pseudo heptolose, and we're going to see this inshallah in the pentose phosphate pathway. Also nonose is like neuraminic acid, etc. As you remember, also sugars can form isomers, so and different types of isomers actually. So let's take the glucose, like the parent molecule of these monosaccharides or hexoses. This is the orientation and distribution of the different groups, including the hydroxyl groups on carbon number 2, 3, 4, 5. It's also on 6, but again, this is a chiral center. Carbon number 1 as well as a chiral center, if you remember. If If we change the orientation of OH group just on carbon number 4, this results in the formation of another type of monosaccharides called galactose. And remember that the relationship between them as stereoisomers is epimers. So they're epimers because they're just different in one chiral center. They're exactly the same except on carbon number 4. So they're C4 epimers. If we change this orientation just on carbon number 2, so this creates mannose, again, another epimer, but on carbon number 2. But if we change the position of the carbonyl carbon, instead of being on the first carbon, becoming on the second carbon, this creates fructose. So these are isomers, but they're not epimers, okay? Or this is a type of stereoisomerism, but it's not epimer, as we've seen in mannose and galactose in relation to glucose. Remember also we discussed another type of stereoisomers called enantiomers, and this is actually found between the same type of molecules. So for glucose, we have a D-glucose and L-glucose. For galactose, the same. We have a D-glucose and L-galactose. The difference in this situation, or this type of isomers, is created by mirror imaging of the molecules. So all of the OH groups, as you can see in here, were reversed. So on this side, this one was on the left. Now the first one, on carbon number 2, it was on the left. On carbon number 2, on D-glucose, it's on the right. On carbon number 3, right, left. Carbon number 4, left, right. Carbon number 5, left, right, etc. So they're exactly, or they are non-superimposable mirror images. So as you can see in here, it's like a mirror, and we superimposed everything. So this creates two different enantiomers of the same molecule. It's different than the examples we discussed, or the type of isomerism we discussed previously, because it's created different types of molecules. Fructose is different than glucose. Galactose is different than glucose, etc. But now we have D-glucose and L-glucose. And in our body, if you remember, we only use D-glucose. Another type of isomerism is is created once these different types of monosaccharide form a ring structure, and this is highly probable. And actually, these molecules are found in ring structure much more than the linear structure or open chain structure. So as you can see on the left side, we can see the first ring that can form. And again, the OH groups that were on the right side are going to become downwards, and those on the left side become upwards of the ring. So the two ring structures on the left and on the right are exactly the same, except on the OH group on carbon number 1. And remember that carbon number 1 in the linear structure was actually a carbonyl, but the formation of ring structure converted it back to an OH group. So this OH group has some sort of freedom to be downwards or upwards, okay? And it actually alternates between the two states for each molecule. So the molecule can become, if the OH group is oriented downwards, it is the beta D-glucose or glucopyranose, it's the same. It's on the oriented upwards, it's going to be an alpha D-glucose. And remember that the beta glucose is much more, or is more stable than the alpha glucose because of the lower steric hindrance. We have we have two OH groups, as you can see in here. And here the OH groups are in the opposite directions. Okay. Now we have the larger molecules, or once we connect two monosaccharides together, we're going to form disaccharides. Tamam? Disaccharides, if you remember, are connected by glycosidic bonds and different types of glycosidic bonds, depending on the monosaccharides they're made of. So in this example here, you can see this is a glucose on the left side. This is a glucose, and we can tell it's a 6-membered ring, OH group on carbon number 4 and downwards, so it's a glucose. Naam.
آسفة على المقاطعة، بس بالسلايد اللي قبل هيك مش زي كأنه كانوا ملخبطين الألفا والبيتا، إنه البيتا هي اللي فوق أو اتش والألفا هي اللي تحت.
اوكي يس انا حسيت انه في شيء غلط، يمكن عاكسين الاسماء، لازم احط الصور اعكسهم. اوكي anyways، فيس البيتا اللي هي على الرايت سايد اللي هي الاو اتش upwards مش downwards. يس so less steric hindrance، والالفا on the left side فلازم نشقلبهم هدول don't worry. فيس again البيتا مظبوط is more stable because the OH groups are in the opposite directions. أما الألفا هم less stable because the OH groups are on the same direction. Thank you. طيب let's go back to disaccharides. As we said, they are made of two monosaccharides connected by glycosidic bond, different types of glycosidic bonds depending on the types of sugars connected via this link. For example, this mannose on the left side, again, we said this is a glucose, 6-membered ring, OH group on carbon number 4 with downwards, so this is glucose. And the second residue again is glucose, 6-membered ring, OH is oriented downwards on carbon number 4, and they are connected via this alpha linkage because the OH groups that were connected via this glycosidic bond are on the same orientation. They're both downwards, okay? So this is an alpha sugar. This is an alpha glucose, and this is again an alpha glucose, and they're connected by alpha 1,4 linkage, creating maltose. This other example on the right side is lactose, made of the first one, which is galactose. OH group is oriented upwards, and again 6-membered ring. And also we have the glucose on the as a second residue, and we can tell that it's a glucose, again 6-membered ring. OH group is directed downwards. So you can see that here the bond is different. The shape of the bond is different because the OH group on carbon number 4 of glucose is oriented downwards, but the OH groups of galactose on carbon number 1 is oriented upwards. So the linkage is of beta type. So it is between a beta galactose and this glucose. Okay, so this is a beta 1,4 linkage in lactose. This glycosidic bond of whatever type is going to be hydrolyzed, so we need an H2O molecule to break down this linkage, and different types of glycosidases. So for lactose we need lactase. For mannose we need maltose we need maltase. For saccharose, saccharase, etc. Different types of glycosidases. Let's now talk about the digestion of these carbohydrates. So we start by ingestion of different types of sugars. Let's say we ingested starch as a polysaccharide, lactose as a disaccharide, saccharose as a disaccharide, cellulose from plant sources as a polysaccharide. In the oral cavity, there is the oral or salivary alpha amylase. This one acts on alpha 1,4 linkage. So in cellulose, we don't have alpha 1,4 linkage, so nothing would happen. Saccharose, the same. Lactose, the same. Starch has alpha 1,4 linkage, so it's going to cut it at some locations, not all locations, creating maltose, isomaltose, and starch dextrins. Okay, just smaller pieces or fragments of starch. Now this mixture is going to move to the stomach. Inside the stomach, alpha amylase activity is going to stop because of the low pH of the stomach. Then the whole mixture is going to move again to the small intestine. In the small intestine, there is the pancreatic alpha amylase is going to get released. And this one again acts on alpha 1,4 linkage. So it's going to break the starch dextrins into isomaltoses and maltose molecules. Okay, and And other enzymes are going to act, but they're not of pancreatic origin. We have some mucosal cell membrane bound enzyme. So they're intestinal enzymes protruding from the mucous membranes or cell membranes of intestinal cells, like the isomaltase, maltase, lactase, and saccharase. So the isomaltase breaks the alpha-1,6 linkage. The maltase breaks the alpha-1,4 linkage, and it has to be between two glucose. Again, isomaltase between two glucose as well. This linkage. The lactase breaks the beta-1,4 linkage between galactose and glucose, and saccharase breaks the anomeric 1,2 linkage between glucose and fructose. Okay, now we have our large sugars, polysaccharides, disaccharides, etc., converted to the simple form of monosaccharides, which is glucose, fructose, galactose, etc., and these can be absorbed easily via different mechanisms that we'll talk about later, inshallah. And then they move to the portal circulation. And then from portal circulation to the liver, and liver can distribute them from liver. They can be distributed or used even in the hepatocytes. As you can notice from here that the cellulose as a polysaccharide is going to exit through feces because it cannot be digested. The type of linkage beta-1,4 between glucose and glucose residues found in cellulose cannot be digested. There is no cellulase enzyme in our intestine. That's why we cannot digest it, and it can get out of our bodies. Okay, so let's just have some larger pictures when we talk about the alpha amylase and how it acts on the linkages inside a starch. And this is the starch molecule. This is the main chain. This is one branch. This is the alpha-1,6 linkage here. All other linkages or bonds are actually alpha-1,4. So the salivary and pancreatic alpha amylases act on the same linkage as you can see in here. So it's going to be just the these linkages that can be broken down. So producing maltose as a disaccharide. Say, for example, it's cut here and here. So this is a disaccharide unit. If we cut, for example, here, okay, here, and here, so this produces this disaccharide with two glucose connected via alpha-1,6 linkage, and this is isomaltose. We may have larger pieces with three residues like trisaccharides. We may have even larger pieces like dextrins, which are oligosaccharides with alpha-1,6 branches. Say, for example, it's cut here and here, as well as here. So this piece is considered as a dextrin. So the maltase acts on alpha-1,6 linkage between glucose and glucose. Maltase acts on alpha-1,4 linkages between glucose and glucose, which is maltose actually. Saccharase between the anomeric or alpha-1,2 linkage in saccharose between glucose and fructose. The lactase acts on lactose, specifically on the beta-1,4 linkage between glucose and galactose and glucose. Trehalase acts on the alpha-1,1 linkage in trehalose sugar, and alpha-1,4 linkage of glucoamylose is going to be broken by exoglycosidase. Let's talk a little bit about these enzymes, the intestinal enzymes that act on disaccharides specifically. So we have the saccharase isomaltase complex, okay? So the saccharase enzyme, as you can see in here, and the isomaltase are going to be in the same, or are actually within the same protein or polypeptide chain. So they are synthesized as a single protein that has a transmembrane domain, as you can see in here. So it's inserted in the intestinal cell membrane, and it's protruding towards the lumen. So this side is on the lumen, where the enzymes are found, but the other side, the intracellular side, actually has a very small N-terminus to the inside of intestinal cells. So they were, the saccharase and isomaltase, synthesized first as a single protein, single polypeptide chain. Then they were cut or cleaved into two polypeptide chains. So now we have two subunits, okay? One of them acts on saccharose, which is the saccharase part, and the other one is the isomaltase part, which acts on the alpha-1,6 linkage of isomaltose, okay? Even though the name implies that it only acts on saccharase and isomaltase, this type of complex also acts on maltose, okay? So هذا الcomplex acts on 3 disaccharides: saccharase, isomaltase, and maltose, okay? And actually يعني المالتase activity 80% of its activity is made of or is actually a maltase activity that breaks the alpha-1,4 linkage of maltose sugar, okay? And okay, this is the first complex. Let's talk about another complex. The other complex is the maltase exoglycosidase. Okay, again, this one is protruding to the lumen of the intestinal cells. It has a maltase activity and exoglycosidase activity. So it acts specifically in glucoamylase. But the difference between this complex and the first complex we talked about, that this is a single protein. It stays as a single protein. There's no cleavage and creation, and there's no creation of two subunits as we've seen in the saccharase isomaltase complex. This is again what I've discussed with you and said to you. You can just read it. You don't have to write them. So let's have an application, or let's talk about the third, let's have an application about this complex and then talk about other enzymes in the intestine that act on carbohydrates. If a patient has a saccharase isomaltase deficiency, what would happen to the degradation of these disaccharides, the saccharase isomaltase as well as the saccharose isomaltose as well as maltose? So there are different causes for this to happen. Say, for example, this enzyme complex, as it is encoded by a gene, there is a genetic problem or there is a genetic mutation in this gene. So this would create a deficiency in this enzyme. In some patients with intestinal diseases, there might be sloughing or loss of the intestinal cells, and definitely we would lose the enzymes associated with these cells. In some cases also of malnutrition, it's like a deficiency of the enzyme, or it may induce some changes in the intestinal cells, and this would interfere with the absorption of these sugars. Also, the injury of the mucosal cells, the intestinal mucosa, by some drugs may again damage the cells and again causes a problem or takes these enzyme molecules with them and results in a problem with the digestion and absorption of these sugars. Also, in cases of severe diarrhea, for some reason, there would be no chance for these enzymes to act on their targets or on their substrates, the saccharase isomaltase and saccharose isomaltose and maltose, and they would be just excreted with the feces because of the diarrhea. Okay, so these are the causes. So what happens when this whatever cause is resulting in a deficiency in saccharase isomaltase complex? We would expect that these sugars or disaccharides are going to accumulate in the intestinal lumen, and this would increase the concentration of the solute in the lumen, intestinal lumen, resulting in changes in the osmotic pressure. This change in the osmotic pressure will increase the movement of water molecules to the intestinal lumen to reduce their concentration, resulting in diarrhea. Okay, as well as or other also symptoms resulting from the accumulation of these disaccharides, that they can be used, these disaccharides can be used by intestinal bacteria, the normal flora, and the side products of their metabolism can cause some bloating, gases, etc. يعني بتصير عندهم نفخة بالإضافة للإسهال اللي حكينا عنه قبل شوي, diarrhea. Let's talk about another enzyme that also protrudes from the intestinal cells and can digest the another type of disaccharide, which is the lactase. Lactase is responsible for digestion of the beta-1,4 linkage between galactose and glucose in lactose sugar. Okay, so this enzyme reaches the maximal activity in the small intestine by 1 month of age, because, as you know, the babies or newborn depend mainly or depend just on milk as in their diet. So they have to have a good activity or a high activity of this enzyme to digest the milk they consume. But this activity is going to decline with age, and it's going to reach the adult level at 5 or 7 years of age. يعني بصير واحد عمره 5 سنين ل7 سنين بتبطل الاكتيفيتي زي ما كان baby. You can يعني eat other types of food and you can digest them with other enzymes. Okay, and this activity is around 10% of the infant level, اللي هو بيعتمد فعليا بس على المilk as a diet. Many people get this enzyme reduced in level, يعني الكمية or amount, or reduced activity of the enzyme, depending, but the result or the end result is the same, that this enzyme is reduced or activity is reduced, resulting in a problem in digestion of lactose. Okay, طيب شو بيصير نتيجة لهالكلام؟ If there is no digestion of lactose, as you can see in the figure, there's no produced galactose and glucose. This would accumulate the lactose in the small intestine, and the intestinal bacteria is going to use it for its metabolism, producing different side products like CO2, methane, and other types of gases and molecules that would result in bloating. As well, and the same thing that happened in the previous type or previous example we discussed is going to happen here, where the accumulation of lactose can be or can increase the osmotic pressure, resulting in withdrawing water molecules towards the intestinal lumen and resulting again in diarrhea. Okay, so you can imagine how big the problem by, for example, these simple numbers downwards, where just consuming 1 cup of milk in an adult, which contains around 9 grams of lactose, is going to result in the loss of 1 liter of extracellular fluid due to the water movement to regulate or to lower the concentration of lactose in the intestinal lumen and adjust the osmotic pressure. طبعا هاي المشكلة is very common لدرجة إنها صارت تعتبر الطبيعي يعني، ففي نسبة كبيرة يعني من مجتمعنا، وعشان هيك الناس بتلاقيهم بقول لك أنا ما بحب الحليب، الكبار يعني، حتى أنتم في منكم حاليا بقول لك أنا ما بحب الحليب due to this problem or due to these symptoms that result from consuming lactose found in milk. طبعا يمكن we probably discussed this before. The solution is to reduce consuming milk or consuming milk with with lactase enzyme or without lactose, etc. So we can reduce this problem in these patients if they want to consume milk. But they can rely, for example, on other dietary, on other sources for calcium. They can eat yogurt, they can eat cheese, etc. These would not cause the problem associated with drinking milk. يعني هي المشكلة مقصورة أكثر بالحليب نفسه أكثر من منتجات الحليب أو مشتقات الحليب. مرة تانية هون عم بيفرجينا كيف اللاكتيز deficiency بتعمل. This is again the lactase deficient cells. This is the intestinal lumen, there is no absorption into the cells. Again, lactose can be fermented by bacteria, resulting in زي ما حكينا side products that cause bloating. It also changes osmotic pressure, withdrawing water molecules to the lumen. And as we said, إذا في عندي 9 grams يعني شرب له كاسة حليب وصار في عنده 9 grams accumulating in the intestinal lumen, this would withdraw 1,000 mils يعني 1 liter of extracellular fluid راح يجيبه لهون، وبالتالي this would cause distension، راح يعمل زي يعني مط أو الgut wall أو الintestinal wall. This would activate peristalsis اللي هي الانقباضات اللي بتصير في الأمعاء، عشان هيك بصير في عندهم diarrhea. This would result طبعا this would take all the components and molecules found in the intestinal lumen. يعني ما راح يطلع بس اللاكتوز، راح كل شيء موجود أكلته أنت مع هالكاسة الحليب مثلا، بعد ما شربتها أكلت معها أشياء تانية، كله راح يتضرر من البيرستالسيس هذا اللي صار، وراح يطلع برا الجسم، فبالتالي بصير في عنا malabsorption of different types or other types of molecules like fats, proteins, drugs, etc. Okay. خلينا نحكي هلا عن عملية الabsorption absorption of sugars و of monosaccharides specifically. في عنا different ways. There are different ways for absorption of sugars, and they need to have transporters because they are polar molecules. They're relatively large, so we cannot just rely on them to diffuse, or we cannot rely on diffusion through the membrane, so we have to have transporters. The first type of transporters is a sodium independent facilitated diffusion transport system or transporters. These are present on the cell membrane, as you can see in here. This is the outside, this is the inside of the cell. So they bind these monosaccharides on the outside of the cell. This would induce conformational changes that closes the molecule from the outside, okay, and open it from the inside and allow the entry of these monosaccharides like glucose into the cells. These are abbreviated as GLUT, yani like glucose transporters, from number 1 to number 14, and they have different characteristics, and we'll take some examples in a minute. As we said, this is sodium independent, so nothing to do with sodium. It's a facilitated diffusion mechanism, so it doesn't need energy, and this is important. And this is also logical because we have high concentrations, for example, on the intestinal luminal side, in comparison to that in the inside of the cells. So this would move them easily. These also can be found on the intestinal brush border. Okay, as you can see, these ones, the pink ones, on the intestinal brush border, and they also can be found on the basolateral surface to allow the exit of these molecules to the capillaries, to the portal circulation. The other type of transporters is sodium-dependent co-transporter system, or the SGLT. This one, as you can see in here, is present on the brush border, for example, of the small intestine for active uptake. For example, if the concentration is low of glucose and galactose, etc., we would absorb them through this type of transporters on the intestinal cell brush border. They're also found in the kidney for the reabsorption of sugar and the avoidance of excretion of these molecules into the urine. So they're found in the proximal tubules to reabsorb glucose back to the circulation and get it back to our cells. Okay, so since it is against concentration gradient, it needs energy. And as we said, it's used under certain conditions where we have lower concentration of glucose from the side in which it's found in comparison to the site to which it is moving. There are different or several examples, and we'll highlight them as you can see in here, and distinguish between the different types of transporters. حكينا في عنا GLUT 1, 2, 3, 4 until 14. We'll just take some examples on these. So for example, GLUT number 1, this one is or has a high affinity to glucose, okay, and it's associated usually with the cells that act as barriers. So there are different barriers like the blood-brain barrier, there's the blood-placental barrier, blood-retinal barrier, blood-testis barrier. So this type of transporters is specifically found in these tissues and these cells. The second type, the GLUT2, this one is non-specific. Okay, so it can transport glucose, galactose, fructose. It does not distinguish between them. It's found in several tissues, specifically the active ones, the liver and kidney. And as I said, they have low affinity and high capacity. So they transport many molecules of glucose, galactose, and fructose to these different types of cells. GLUT number 3 is the major transporter. It's transporter in the central nervous system with high affinity. And remember that the brain rely mainly on glucose as a source of energy. So it has this specific one to guarantee that enough glucose is reaching to brain to supply it with the needed energy. The other or the next type, GLUT number 4, this one is the insulin sensitive transporter. So once you, for example, ingest large amount of sugar or ingest after a meal, there would be secretion of insulin to take this glucose in the meal from the that was absorbed after digestion of that meal, take it from the or increasing its uptake from bloodstream and uptaking it into the cells. So it is sensitive, or it's going to act and its expression is increased after insulin secretion is increased, yani after a meal. The other one is GLUT number 5, and this one is important for the transport of fructose. So it's not a glucose transporter, it's a fructose transporter, and it's found in cells that, for example, the intestinal epithelium, because we may have large amount of fructose, specifically if we ingest saccharose, the common table sugar. So this would produce large amounts of fructose, resulting in the need to absorb it through this membrane. And it's also found in spermatozoa because these cells depend on fructose as a source of energy. GLUT number 7 is found in glucogenic tissues. Glucogenic tissues means that the tissues that can synthesize glucose, okay? And it's found specifically at the ER membrane. So how can we compare the transport of glucose in a neural tissue, where we have a blood-brain barrier, versus a non-neural tissue or cells? So this is the neural tissue, the first one on the left side. Remember, there's the blood-brain barrier. So what does this mean? The cells are connected via tight junctions, as you can see in here, tight junctions to forbid the movement of molecules between cells. There is a continuous basement membrane under the endothelial cells that line the blood vessels, again to avoid or to forbid the movement of molecules through this. ما هو البارير يعني عشان يمنع الحركة، فبالتالي لازم نحط زي حوائط صد في وجهنا، وهي ال tight junctions first one، وفي عندي كمان اللي هم continuous basement membrane. Other factors that also contribute اللي هو packing of the cells، يعني there are smaller distances between the cells، يعني ملزقين ببعض أكثر. They also lack penocytosis. Penocytosis اللي بنعرفها هي الشرب الخلوي. This process is not present in these cells. فبالتالي for the transport of glucose, I need to have transporters of glucose both on each side of the cells. يعني this is one side of the cell. We transported glucose through it, through this transporter, and we need another transporter on the other side of the cell to guarantee that this glucose molecule is going to pass to the nervous system and different types of neurons and cells in the nervous system, so they can be supplied by this glucose. Whereas in non-neural tissues, as you can see in here, the distances are larger between cells. There are no tight junctions, as we've seen there. The basement membrane is non-continuous, so this would allow the movement of glucose through different ways, either between the cells, okay, diffusion, okay, between the cells, or through the transporters, as you can see in here. So they can move easily than the other types of, or the neural tissues or neural cells. Penocytosis is also active, as you can see in here. So what happens when this glucose is, or after absorption of glucose into the bloodstream? We said that it moves to the portal circulation, and from portal circulation to liver. From liver also it's going to go to systemic circulation. But it should not stay in the bloodstream, because if it stays in the bloodstream شو بصير عندنا؟ بصير عنده سكري فعليا إذا ضله. The concentration of glucose becomes very high. It cannot be used by cells to produce energy. It's going to withdraw water molecules towards the bloodstream. فيعني بصير في لها آثار كتيرة. We have to move it to cells so they can make use of it. How does this happen? By increasing the transporters on the cell surfaces, as you can see here, specifically the active tissues like muscle, adipose tissue, liver, etc. How can this happen? By, as we said, secretion of insulin. Insulin is going to bind to its receptor. The binding of receptor is going to activate the signaling pathway through a cascade of events that would increase the expression and synthesis of glucose transporters and the movement of these transporters to the cell membrane. So their concentration increases and the transport of glucose is going to increase through cell membrane. The last thing for today is once we have this glucose reaching the cells, as we discussed in the previous slide, this glucose is going to get metabolized. So we're done with the digestion, absorption, distribution, and now the last thing is metabolism. Metabolism يعني أكسر هذا الجلوكوز into a smaller molecules and extract the energy stored in the chemical bonds of glucose. There are different and several pathways in which this glucose can be consumed. The main one اللي هو راح يدخل في عملية glycolysis to pyruvate, والpyruvate بيصير acetyl-CoA and then into Krebs cycle. It may also produce lactate إذا مشينا في الanaerobic pathway. This lactate and even the pyruvate can be used to synthesize glucose in gluconeogenesis in case we have low glucose concentration. يعني this is the reverse pathway for glycolysis. The gluconeogenesis, gluco يعني glucose, neo يعني جديد, synthesis جديد, genesis يعني formation or synthesis يعني تصنيع جلوكوز جديد from sources other than carbohydrates. يعني ما بدي أجيب مثلا starch وأكسره وأعطي منه glucose. Other pathways also, when we have excess glucose, more than the need of the cells as a source of energy, it can be stored in the form of glycogen. Glycogenesis is the process or the pathway. Once we need this glucose stored in glycogen, we're going to break it down via glycogenolysis, يعني glycogen lysis or تكسير glycogen to produce glucose. This glucose again can be used to produce pentoses or other types of sugars and NADPH that's needed for other reaction as a reducing power. اسمها الباثوي pentose phosphate pathway, and it also can be oxidized to glucuronic acid, etc. for use for other purposes in our cells. Okay, so this is just an overview, and inshallah we'll go into these pathways one by one in a logical sequence of lectures. راح نبلش إن شاء الله next time with glycolysis, this first pathway, and inshallah we'll continue with the other pathways later on. Thank you all for your attention, and we can take questions now. خليني أرجع. أشوف هلا مين بده أو مين في عنده question. مين عنده سؤال؟ سامعيني؟
أنا حابة أحكي. هلا هو مو سؤال على قد ما إنه يعني حابة أطلب منك شغلة. أول إشي يعطيك العافية.
الله يعافيك.
تاني شغلة دكتورة، إحنا حابين إنه تكوني شوي أبطأ بالشرح، لأنه يعني كتير أحيانا الواحد بيكون كتير مركز معاكي بعدين بشت.
يعني أوكي. أبطأ من هيك؟ أنا بحاول إني شوية أكون يعني أشد على الكلام عشان ما أسرع. تمام. طيب أنا عندي فكرة تانية لأنه ممكن هاي موضوع السرعة تكون نسبية، يعني ما أعرف أقرر كتير إنه أنا فعليا السرعة المناسبة ولا لا. هلا أنا رح أبطئ شوية، لكن رح أرجع كمان أضلني، ورح تلاحظوا هذا الإشي أصلا لحاله حتى لو المحاضرة وجاهية أنا كنت أعمل هذا الإشي دائما. رح أرجع أعيد الشغلات. تمام. ارجع اعيدها، فهذا بيعمل عمليه زي تاكيد للمعلومه. لو انت هيك شتتي لك نتفه وراحت عليكي شغله بنرجع بنعيدها. فانا دائما راح اكرر. خلينا نعتمد هاي الاليه على اساس
دكتورة هيك
انه
أحسن، وحتى لو مثلا تصير تسألينا أسئلة بعد كل إشي، يعني هيك بنكون مركزين معاكي أكتر.
طيب تمام. هلا جزء كبير من محاضرة اليوم خلينا نحكي قديم بتعرفوه، بس احنا لازم عشان هيك نرجع تو ريفرش يور ميموري. طيب خليني هيك اسال اسئله، بما انه فيش مش شايفه حدا رافع ايده. خليني انا اساله، بدنا متطوعين
دكتورة.
يجاوبوا. مين؟ ما
دكتورة.
شفتش اصبع حدا طالع او
دكتورة,
مرفوع.
in the lactose internus the activity of the lactase decrease or the enzyme itself decrease.
هلا depending، يعني أنا قرأتها في أكثر من text book. Some text books say it's enzyme deficiency، يعني the amount of the enzyme is reduced. Others says إنه it's the activity of the enzyme. بس الأكثر بيحكوا إنه الاماونت. تمام. فأنا حكيت الحالتين لأني شفتهم أو قرأتهم في أكثر من text book هيك وهيك، فحكيت لكم الشغلتين. بس mostly أو خلينا نقول اللي بتلاقيه أكثر اللي هو الاماونت.
The alpha 1,2 linkage is the same as the alameric 1,2 linkage.
في السكروز يس اتس ذا سيم. في منهم بعض التكست بوكس اجين بسموها الفا 1 2، في منهم بسموها انوميريك 1 2. يس السكروز تحديدا
كمان.
يس.
بس آخر سؤال.
تفضل.
هسا in the lactose internus we can it is specifically for the milk, like if we eat cheese. Like we can say that the causes of the process of formation of the cheese can introduce the lactase internus.
هلا المنتجات او مشتقات الحليب بشكل عام، سواء بنحكي عن لبن، لبنه، جبنه الى اخره، هدول كلها منتجات. يعني ما اخذتي انت البرودكت دايركت اللي هو الحليب اللي من البقر ولا الغنم ولا وات ايفر وشربتيه مباشره بعد الغلي طبعا. لا، صار في تصنيع، عمليه تصنيع، وهي عمليه التصنيع بتاثر على الاستراكشر الموجود، فبالتالي ما راح تعمل مشاكل. وهذا الشيء بينتج انه يعني الواحد بتضايق من الحليب بس ما بتضايق، بياكل جبنه ولبنه عادي ما في مشاكل. فهذا بيكون another source of calcium. أنا إذا بتلاحظوا أنا حكيت تحديدا عن الكالسيوم، بس إذا بدنا نحكي عن فيتامين دي اللي الناس كمان تهتم فيه يعني عنصر غذائي مهم موجود في الحليب ومشتقاته. هلا فيتامين دي الكميات اللي بنحصلها من الحليب وحتى كمان من الأكل كله، يعني خلينا نحكي إجمالا قليلة. يعني لو أنا فعليا بدي أعتمد على الفيتامين دي اللي بحتاجه لجسمي من الأكل، بدي أشرب لي كل يوم 10 لتر حليب، وهذا الإشي مش منطقي. يعني مستحيل حدا حتى لو ما عندوش مشاكل في الحليب إنه يشرب أو يعني يسوي. فنتيجة لهالكلام لازم نعتمد على الفيتامين دي اللي هي عملية التصنيع اللي في جسمنا بتصير، اللي هي من خلال التعرض لأشعة الشمس واللي هي فعليا اليو في لايت اللي بياخده الواحد من الشمس راح يكون زي عامل مساعد أو منشط يعني أوائل الخطوات في الباثوي تبع تصنيع الفيتامين دي من الكوليسترول، اللي هو الكوليسترول موجود عندنا وبكثرة وجسمنا بيقدر يصنعه كمان. تمام. فيس البروسيسنج لليوغرت للتشيز اتسرا ما يعني بيخليهم انهم سيف وما بيسووا مشاكل. هلا في لاكتوز انتولرنس بصير عند البيبيز، في ناس عندهم مشاكل باللاكتوز بالحليب منه هو بنولد فيها يعني. تمام. فهون المشكلة مختلفة. تسمعوا أطفال عندهم حساسية الحليب، فهدول لهم different formulas. يعني الفورمولا اللي هي الحليب الصناعي، يعني بنشر لهم أنواع مختلفة من الحليب مناسبة للوضع اللي عندهم. أو إذا ما بدهم يعتمدوا على الحليب الصناعي بدهم يعتمدوا على الحليب الطبيعي، الرضاعة الطبيعية، بتلاقي الأم الطبيب يعني بيعمل زي جدول للتغذية للأم. فممنوع الأم تشرب حليب ومشتقاته، وبالتالي ما راح يطلع في الحليب تبع البيبي، سبحان الله هو حليب لكن ما راح يطلع فيه اللاكتوز، وبالتالي بيقدر إنه الطفل إنه يرضعه بطريقة آمنة وما يعمل له مشاكل. يعني بتنجح بنسبة كبيرة من الأطفال، وفي ناس لا ما بتزبطش، بيضطروا يروحوا على الفورميلاز أو الحليب الصناعي. ومرات هي بتكون هاي الحالة مؤقتة مرات، يعني مرات الطفل بده يتراقب طبعا أوفر تايم، ممكن بعد 2 إلى آخره ترجع أموره طبيعية، ويرجعوا أهله يعملوا له انترودكشن لكل أنواع المشتقات الحليب شوي شوي، يرجع يشرب حليب عادي. فمرات بتكون حالة مؤقتة، ومرات لا بتمتد معه طول العمر، فبالتالي هو مضطر إنه ما يشرب، يعني خلاص عنده تحسس من الحليب، فبالتالي بيبعد عن المنتجات اللي فيها لاكتوز.
We have the maximum activity of the lactase when we are babies.
يس، لانه احنا هذا الوقت على عمر الشهر، يعني طبعا مش انه ما فيش، هلا بنولد الطفل عنده اكتيفيتي مع شرب الحليب، هو رح يبلش ايش يتغدى، بس حليب، فيش عنده غير هذا المصدر الوحيد. فبالتالي بتصير ايش، تزيد الاكتيفيتي، بتوصل الماكسيموم على الشهر، بعدين خلص بتضلها الماكسيموم لحد ما تبلش تتناقص وتوصل المينيمم تبعها، اللي هو نفس يعني بعمر 5 إلى 7 سنوات. فأنت بتوصل المينيمم في عمر 5 إلى 7 سنوات، وبضل هذا الليفل، مش إنه بروح صفر طبعا، لا يعني إنه بتصفر، لا بوصل لحد أدنى اللي بضله معك في الأدلت هود. يعني طول ما أنت أدلت في عندك نفس الليفل تبع أبو ولد عمره 5 سنين أو 7 سنين، أو أنت وعمرك 5 سنين نفس اللي موجود عندك as an adult. فهون في variation، في ناس بضل الليفل عندهم أعلى، فبالتالي عادي بيشربوا حليب ما عندهم مشاكل حتى هم أدلتس، وفي ناس لا بيكون الليفل عندهم أقل، فبالتالي بصير يقول لك خلاص أنا ما بحب الحليب، ما بكرهه، بتضايق منه. بتسمعوا الكبار بالذات، أو في منكم ناس بقول لك بس أشرب حليب بصير عندي نفخة، بصير في عندي إسهال، بصير في عندي شيء، فبتضايقوا منه. وزي ما حكينا يعني it's consistent with adulthood، يعني بتمشينا في الادلتهود، ما في مشكلة، لأنه خلاص فعليا النمو خلاص وقف. بنحتاج للكالسيوم لعظامنا. فيتامين دي بناخده من مصادر تانية. فيتامين دي زي ما حكيت السينثيسيس أهم من الاعتماد على الأكل بشكل عام، مش بس على الحليب كمصدر له. فيعني consistent مع الوضع تبعنا، مش إنها يعني حتخلق لنا مشكلة كبيرة، لأنه أصلا هي فعليا very common، يعني أكثر من نص الناس في العالم بصير عندها هاي الديفيشنسي مع العمر، فصارت تعتبر هي كأنها الوضع الطبيعي. يعني انتوا هلا اقعدوا مع الكبار او اقعدوا مع بعض احكوا، النسبة الأكبر ما بتحب الحليب أو بتتضايق منه أكثر من اللي ما بتضايقوا، فصار يعتبر كأنه هذا الطبيعي يعني.
دكتورة أنا بالميتابوليزم ما بيهمني إذا كان هو DRL صح؟ بس إذا صنعت DNA
لا هلا فعليا في الميتابوليزم احنا يعني خلص مش هنقعد نحكي دي وال، لكن هو فعليا اللي بيشتغلوا عليه الانزيمات هو دي او ديف عفوا ال، لا الشجر الناتشورال الموجود عندنا جوا جسمنا هو الدي، وهو اللي بيشتغلوا على الانزيمات. لكن في بعض الاحوال ما يعني طبعا بيعتمد على الباثوي وبيعتمد على الشجر اللي بحكي عنه، الالفا والبيتا بتفرق، وفي بعض الاحوال ما بتفرق. يعني مثلا الجلوكوز
بس
لو بدي ادخله
بالتصنيع رح يفرق معي، لأنه أنا بدي DNA، مش DNA بيكون compacted.
حسب ما بقول لك لسه انت انا ما كملت لسه. هلا اذا بحكي عن جلوكوز فقط وبده يدخل في عمليه جلايكوليسيس اكسره عشان يحصل منه طاقه، لازم يكون دي. لكن ما بفرق عندي الفا او بيتا، يعني رح يتعرف عليه الانزيم سواء الفا او بيتا ويدخله ويكسره ويعني نستفيد منه. في حال بحكي عن رايبوز او 2 دي اوكسي رايبوز تحديدا، يعني بده يدخل في تركيبه الدي ان اي، فهذا يس لا بتفرق الفا ولا بيتا، لانه مش لحاله موجود الشجر، هو راح يكون ضمن يعني هيوج موليكيول اللي هو جزيء الدي ان اي. وجزيء الدي ان اي زي ما حكينا جزيء ضخم جدا، فيه 3 بليون بيز بيرز، وهذا الرقم كله محصور في نواة الخلية، ونواة الخلية فيها أشياء تانية مش بس الدي ان اي، فبالتالي it has to be compacted. جزء يعني من أهم الأجزاء من الكومباكشن، كونه بنحكي إحنا عن عدد هائل من النيوكليوتيدات مكررة، فإذا كل وحدة هيك أعطتنا سبيس صغير زيادة أو سهلت لنا الكومباكشن شوية، عدوها ضرب 3 بليون، فبالتالي إيش راح تسوي عندي؟ راح تأثر بشكل كبير على عملية الكومباكشن. فلازم تكون بنوع أو بشكل محدد. بس هو دي طبعا دي رايبوز، بس الفرق بين ألفا وبيتا واحد منهم فقط كان بإنكوربوريتد، الثاني ما بنقدر، لأنه ساعتها الكومباكشن راح يتأثر. طيب مين في كمان عنده سؤال؟ الإشي الجديد هون احنا يعني شوي الدايجستشن والابزوربشن هدول اللي خلينا نحكي المعظم الجديد تركز فيهم. فهمتوا الابزوربشن different transporters، إشي سبيسيفيك وإشي مش سبيسيفيك، إشي سبيسيفيك للتيشو، إشي سبيسيفيك للموليكيول، إشي منهم مثلا زي ما حكينا صوديوم ديبندنت، إشي صوديوم اندبندنت، إشي بيعمل لنا اكتف ترانسبورت، إشي فاسيليتيتد ديفيوجن. يعني بيعطينا هيك وايد فرايتي اوف تشويسز. وبالنهاية راح يقدر يوصل لي كل أنواع المونوسكرايز اللي ممكن يعني تدخل إلى الجهاز الهضمي، وبالتالي أحتاج أعملها absorption، سواء فركتوز ولا جلوكوز ولا جالكتوز ولا. طيب مين كمان عنده سؤال؟ تمام هيك خلاص أمور منيحة. طيب معناته موعدنا إن شاء الله يوم الأحد القادم الساعة 12